So, welcome class. Welcome everyone who is joining us. A very good evening. Warm welcome. I know it's very hard. These terms like organoids, embryoids, organogenesis, embryo culture, anther culture, pollen culture, wide hybridization, pathogen eradication, germplasm preservation. Suddenly, you all had a huge amount of terms coming up to you. Understand they are quite hard to digest and to go through with them because this is all quite subjective. Until and unless you see the practical part of it, things will be always hard for us to understand. So to do so, we will I'm trying to bring them into the practical part so that you can whatever we are learning here, we can also learn the practical part of it. So in that case, I had thought of doing some practicals with you. I have practicals PDFs with me, which I was teaching to students. So some of the practical from the syllabus we can do it here also, like gel electrophoresis, plant tissue culture, lab DNA extraction. All these will be doing. Beyond that, other ones like production of disease-free plants or micropropagation, cell suspension culture. I can teach you mainly subjective how they are done in the lab. What are the main requirements, main media do you require while doing so? So that when you are leaving this workshop, you are not leaving only the subjective part but also with the practical part. So that's I always insist while teaching that if you're learning anything, you should also be good in words with the laboratory part also. So I will take care of that. Don't worry, don't worry. So today we will be dealing with the in vitro tissue culture, and later part was second part was micropropagation. Yes. So we'll be dealing most of the techniques today, like anther culture, pollen culture, ovule culture. So these things we'll be dealing today, all various cultures, and also besides them, we'll be dealing with the micropropagation also. What are the various steps in micropropagation and what are the various applications of it? So all we'll be dealing with that today. So please keep joining in us. I will try. I mean, so to record the sessions, let me see if I can turn on the YouTube besides this. If not, we continue like this. So let's see after the session if things are good. I will share with you one link where you can have the things if it is keep going fine. And I will share your link with you afterwards. No worries, no worries. So let's join in everyone. Okay, I will try to make some YouTube session if possible. I will send it later. So it's 8 p.m. So let's wait for another 5 minutes. Then we start our lectures. So how was the students' weekend? Uh, let's have some interactive chat. How was it? What you did it? Did you cook for yourself? Cook for your family? Went outside? Did your hobbies? Have some relaxed time? Doing assignments, homeworks, college time? So how was the weekend all in all? Mhm. Great, great. Weekends are meant to be enjoyed, to do your hobbies, to do your stuff, to get relaxed. Okay. And others, how was the weekend? Was good? So, anyhow. All is good. So I think we can start now. We had enough students. I'm trying to record the sessions via YouTube. I don't know for how long it will go, but in meantime it's recording. But I am not checking it, so I will be concerning with the slides. If it's get recorded, then yeah, just subscribe to my channel and check it there with the earlier videos. So if it is uploaded, then it will be. Otherwise, sorry. So I'm trying to record it. Okay. So your videos are being recorded on YouTube already. So if everything went well, if there is no breakout happen in between, then it's all good. Otherwise, yeah. So let's start. Just wait 1 minute more, maybe. So we start at 8:05. Okay, okay, we understand that situation. No worries. So, okay, let's start the session. Today, we will be dealing the next part of your syllabus in the unit 1, that is techniques of in vitro culture. So we'll be dealing with various techniques from your organ culture to your endosperm culture. Various cultures we'll dealing today, mostly covering your unit 1. Actually, we might be finished by unit 1 by today on the third day itself. And yeah, it's good to manage things within stipulated time. So as we know, we have discussed a little bit about tissue culture before, but now we go in more details about it. Um, so anyone can tell me what is the definition of redifferentiation? Let's open the chat. Anyone can tell me the definition of redifferentiation? Yesterday we did that. Cell mask as specialized cell type. Okay, not so nearby. Yeah, but I'm trying to record the session, so don't worry. Yeah, differentiation is occurring in the undifferentiated tissues, right? Yes, differentiation, undifferentiated tissues, that is redifferentiation. So that's the main thing. and differentiation is occurring in the undifferentiated tissues. Yeah, that's the redifferentiation. So a little bit. So this is your now today fifth lecture, I guess, from your series of plant biotechnology. So where we have done all the basic history, scope, and totipotency morphogenesis part in quite detailed manner. Now today we will be dealing in detailed manner with the techniques of in vitro culture, micropropagation. These part will be automatically covered in your in vitro culture. Besides your test tube fertilization, that we will do on another day. But we will be handling with all other parts positively. And then second part we'll be dealing is the micropropagation. So all these things will be dealt slowly and easy manner. So let's start. So we will deal in this techniques of in vitro culture. Um, your various features of in vitro culture, some important terms, then tissue culture techniques such as and their importance also, organ culture, seed culture, meristem culture, embryo culture, ovary culture, anther culture, callus culture, cell suspension culture, protoplast culture. So there are kind of 9 types of culture that we'll be dealing. Um, and their various applications, various importance, and how and where they are being used. So these topics, all your these types of culture, they are dealing here your topics of theory. So I have to add students, please come on time. Okay. So we have discussed yesterday. A day before yesterday, actually, on the first day of our lectures, that plant cells divide indefinitely in a appropriate culture medium and regenerate to whole plant. And tissue culture refers to the growing of living plants, tissues, or organs on suitable nutrient medium in sterile conditions. For in vitro culture, explant from healthy and young plants of the plant is used to regenerate the whole plant. So in vitro culture could be done in the test tube, dish, or flask. That are the important features of the in vitro techniques, or outside the living organism. That is, we are doing it outside the living organism, in this case, in a test tube, like cloning of test tube baby. Instead of doing all these human things that we were discussing, we are now doing with the plants actually. So these in vitro techniques, doing growing your plants in a petri dishes, in a flask or test tube, is referred to as a plant tissue culture. Tissue culture, and in this case it's refined to plants. They're grown with a nutrient medium, and tissues grown are outside the parent organism from which tissues have been taken. So these we have discussed already yesterday: differentiation, de-differentiation, redifferentiation, regeneration. So again, I have put up these terms because they are one of the important terms in remembering the things of the plant tissue culture. So a little bit repeat from yesterday. So differentiation is defined as the process by which meristematic cells are converted into two or more type of cells. Meristematic cells, they are divided into two or more type of cells which are different from each other. And de-differentiation is to denote the process of formation of unorganized tissues from a highly organized tissue. A unorganized tissue is formed from your highly organized tissues. Whereas the redifferentiation is the de-differentiation cells lose the capacity of division and become mature to become specific functions. So these cells are tend to be known as redifferentiated and termed as redifferentiation. And regeneration is forming of entire plant from the explants via callus or directly. And organogenesis that we have done yesterday, this term also, organogenesis is the formation of adventitious roots or shoots from the cells, that is from one of the organ parts of the plant, and it's called as organogenesis. And totipotency is the phenomenon of regeneration of plant to develop into whole plant, whereas the embryogenesis is the starts from the single embryonic cell, that could be zygote or undifferentiated callus cell. Both of them could go for embryogenesis. So embryos developing from zygotes are called zygotic embryos, while the developed from the other cells other than the zygotic cells, that is somatic cells, they are called somatic embryos. So hence we have somatic embryogenesis and zygotic embryogenesis. So in this first case, your apical meristem cells, they are producing dermal tissues, ground tissues precursors, and also vascular tissues precursors. And then further, they are going for pavement cells, parenchyma cells, and vessel elements. And they then go to the making of trichoma, collenchyma cells, sarcoma cells, sieve tube elements. So how you can see how in differentiation each cell type, so they divide into different functions of the cells and different part of your plant, how they're differentially dividing. Whereas in the dedifferentiation from the useful part, we have a dedifferentiated cells as in the form of callus, and then from the callus, the redifferentiation happens. Then you become your organ. From the callus, your organ formation happening, and that is known as your redifferentiation. So that's how these three terms are somehow yeah, interconnected also. And regeneration, as we see in the first lecture, first slide, that if you take the explants and you grow in the petri dish, or take the isolated cells, grow in the agar medium, and then you have your first shoots, then roots, then you go for your in the soil culture, then you have your full plant. That is your regeneration. Embryogenesis is something with the embryos where it goes with the zygote cells or the somatic cells, giving you a heart, torpedo, triangular, globular, ocant, and two cell various stages of embryoids, and at the end you have a mature embryo for your further functions. And this is the organogenesis started from your callus. From callus, the shoots are started to happen. Then from the shoots, the roots are happening. So various organs are being produced with the help of organogenesis. So that was a summary of what we did so far in nutshell in last 2 days within these 3 or 4 slides. Then tissue culture technique. It's an in vitro technique which give rise to new plant by regeneration of tissues from and organs in the nutrient medium. So there could be various plants that you could use, parts of the plants you could use, that is meristem, embryo, anther, cell, ovule, all these could be used for regeneration. So plant tissue culture could be divided into further paths based on the paths that you're using. It could be meristem culture, protoplast culture, cell culture, bud culture, organ culture, callus culture, embryo culture, seed culture. So as you take the part of the plant, so that will be your further culture will be known as. If you're taking two protoplasts, then it's a protoplast culture. If you're taking cell suspension culture, then it's a cell culture. If you're taking bud, then it's a bud culture. If you're taking some organ of the plant, then it's an organ culture. If you're taking callus, if you're taking embryo, if you're taking seeds, so they are divided into different tissue culture parts. This is as simple as written there. So here you can see how from the adult plant you have taken four different parts of the plants. One is showing you embryo culture, second is the organ culture, callus culture, cell culture. In the embryo culture, uh, you take the embryo from the adult plant, yeah, then you put it in the test tube, then with the help of proper giving them hormones and medium, you grow into new adult plant. Same goes with the organ culture. You take the organ, process goes like callus the same. Whereas in the cell culture you do in the petri dish, then hence you have your first cell suspensions, and then from there you can grow your whole new adult plant. So this is how all four different categories has been shown you how it is done as basic principle. So let's start with the first part of the tissue culture, in vitro tissue culture, that is your organ culture. So culturing isolated organs or tissues such as root, stems or leaf in an artificial media under controlled condition is known as organ culture. Any plant organ can serve as an explant to initiate cultures. So we can use shoot, root, leaf, flower, and in the culture it would be then shoot tip culture called as root culture, leaf culture, or anther, ovary or ovule culture called as. So, what is the importance of organ culture? It provides an excellent experimental system. So it provides a very excellent experimental system to define the nutrients and growth factors normally received by the organs from other plants, other parts of the plant body from its external environment. It's also valuable to study the interdependence of organs for growth hormones and other factors. Beyond that, it's open up new avenue for the development in the agriculture and horticulture. There are two types of organ culture on the basis of the explant, which could be classified as further. You can see as a vegetative organs and reproductive organs. So vegetative organs could be root culture, leaf culture, shoot tip culture, whereas a reproductive organ is a complete flower or isolated pollen culture or isolated ovule and embryo culture. So these are all uh, different reproductive organs and vegetative organs parts of it. So we're done with the organ culture. Next comes your seed culture. Somebody wants to get added. So uh, increasing the efficiency of germination of seeds that are difficult to germinate. So the one which are not able to germinate easily in vivo, but with the help of growth regulators and production of clean seedlings for explants or meristem culture, you can grow them. So here we have shown you an example. Just they have put up the seeds in the required medium, and hence you can grow complete there are plantlets coming out of it. These shoots, these full leaves are coming out of it, and hence you can put them under the further conditions with the required nutrient mediums, and you can grow into whole plant from the seeds. So this is how the seed culture is working out. Then comes the meristem culture. So in meristem culture, you just take some generally a shiny special dome-like structure measuring around 0.1 millimeter in the length. So in the bottom, you can see you take the meristem, which is a shoot tip of your plant. This is the magnified version of the shoot tip, and then you exercise the meristem. You cut it the meristem and put it in the conical flask with the medium, with the culture medium. Then meristem placed on the culture medium like this, and new shoots and callus tissues they started to grow here. Then rooting and condition of transplanted roots, so they are like transferred into the other long edged flask like this one and started to grow. Then you put them into the soil hardened plants, then hence you can have your plants for your for any function that you're looking for. So this is how the meristem culture works like. So it says that the excised meristems can be cultured aseptically on the agar medium, and under appropriate conditions will grow out directly into the small leafy shoots or multiple shoots. Or you can grow some small callus first, then grow into the roots which will develop further. So in this case, cytokinin play a major role as a nutrient medium for the development of the meristems. So example for cytokinin here is BAP, that is 6-benzyl aminopurine. 6-benzyl aminopurine is one of the most effective cytokinin commonly used in the meristem culture. Importance of meristem culture. The uses of meristem culture mainly includes uh, to remove the viruses if there are any. Micropropagation is easy. Storage of genetic resources. These are the three major items, major characteristics that meristem culture is being used. So in the virus eradication, so many plants contain viruses which could be reduced with their potential yield and quality. So it is therefore to produce a virus-free stock which can be multiplied. So generally meristemic tissues are free from viruses due to their high mitotic activity, hence vascular tissues are not developed in the meristemic region. Okay, so therefore meristems are idle explant to produce virus-free stock. So in this case, due to the high mitotic activity, there is very high mitotic activity is happening in the meristemic cells. So that's why they don't have any viruses in them, and therefore meristems are idle to plant virus-free stock plants. In the case of micropropagation, sexual or vegetative propagation of whole plants using tissue culture is known as micropropagation. So meristem of plants can successfully use for the micropropagation. So in the micropropagation, it's mainly the whole plant is being used for the tissue culture technique, uh, taking the part of your sexual or vegetative propagation in that part. So they are also widely used in the meristems are being used in the micropropagation. So few seeds are not separate for storing as a genetic resources. So meristem from such plants can be stored in vitro. So if seeds are not possible to store as a genetic resource, but meristemic part for such plants can be stored for as a genetic resources for in vitro information. So done with this culture also, merismatic. We're done with the first was the our organ culture, then comes the seed culture, then comes the meristem culture. We done some little bit about importance of it. Then comes your embryo culture. The fourth one is now embryo culture. The embryo culture, as we have discussed yesterday also, it's take the embryo first, the embryos, then from the embryos you can grow your whole plant. So embryo of different developmental stages formed within the female gametophytes through sexual process and can be isolated aseptically from the bulk of maternal tissues of the ovule, seed or capsule and cultured in vitro under aseptic and controlled physical conditions in glass containing solid or liquid medium to grow directly into the plantlet. So aseptic excision of the embryo is carried out then, which is transferred to the suitable nutrient medium for development of optimal culture conditions. In case of small embryos, it is important to remove the embryo carefully without causing an injury. The most important aspect of embryo culture is the crucial selection of medium necessary to the sustained continued growth of the embryo. In culture embryos, they are not induced to form callus tissue, but they are allowed to form plantlet. So this is shown you different categories of embryo culture. In the A case, it's shown as a culture of adventitious embryos from the polyembryonic seeds. So from the polyembryonic seeds, so they remove the adventitious embryos of it and then they grow into the test tubes. In the B case is the culture of mature and intact seed embryos. From the seed they are taking this embryo, like in the case of soybean seeds that we saw on that day, the first day of our lecture. Then culture of dissected embryo. So they dissected into different parts. Then culture of immature embryo. So this is the heart and torpedo shape. From the heart we just started to grow. This could be also one of the part for the embryo culture. Then in the E is the undifferentiated embryo of orchid. This is orchid plant. So undifferentiated version of the embryo. We could also grow that part also. Or culture of abortive or inviable embryos. So the one with the plant breeding with the cross, you can also check with those embryos also. So these are the various different embryos, the various different categories of embryo culture, various sources that could use to grow your embryo culture, right? So bache, these all slides, they are highly officially given by the guidelines by the Indian Agriculture Research Institute, IARI. So if these diagrams, if you are studying for plant biotechnology exam or somewhere you have to give for let's say some exam preparation where plant biotechnology is a subject. So these things that I'm telling you here, they are all quite important and might be coming in your exams. So take each diagram, each wordings of these slides, even though they are morely subjective, but they are highly important from your examination point of view of any type, any exam that you're giving, whether it's CSIR, NET exam or GATE exam or yeah, all the examinations related to any plant biotechnology. So these are quite important figures because they have been guideline given by the ARI, Indian Agriculture Research Institute. If you're going for agriculture exam also under AIRI also. So importance of embryo culture, it helps in the analysis of various parameters of embryonic growth. Then they understand the control of the differentiation and nutrition requirement of the progressively small embryos, and they help us to check in the change in the nutritional requirement of embryos at different stages that can be demonstrated by the embryo culture. Now comes the fifth one, that is from the ovule, ovaries, or pollen. Uh, this ovary culture, which is a technique, a culture of ovaries isolated either from pollinated or unpollinated flowers. From the pollinated or unpollinated flowers, uh, the ovaries are isolated, and ovaries grows in culture and form the fruits that ripen and produce viable seeds. So ovaries are grown in the culture and form the fruits that ripen and produce in viable seeds. These development takes place on a simple nutrient medium containing only mineral salts and sucrose. And but the ovaries of unpollinated flowers do not grow in simple nutrient medium, so we use some synthetic auxins such as 2,4-D or 2,4,5-T in the nutrient medium to induce the development of ovaries of the unpollinated flowers. So we need some extra nutrient medium in the case of unpollinated flowers, medium like synthetic auxins such as 2,4-D and 2,4,5-T in the nutrient medium to induce the development of the ovaries of the unpollinated flowers. Otherwise, it's easy to go with the mineral salts and sucrose in the medium. So it is important to study the early development of embryo, fruits, different aspects of fruit physiology, including respiration, maturation, disease, which could be very, very viable to understand, to know the flora and fauna of your plant that you're growing, whether you're growing a strawberry or you're growing cabbages in the field. So to know, to get the desired results of sizes, big sizes of your cabbage or a juicy strawberry, so these ovary culture could be really helpful to understand the um, to understand its physiology. So role of floral organs can be studied from the in vitro culture of ovary. Various floral organs could be studied with that. Um, Furthermore, ovary culture has been used to eliminate the obstacles such as failure of pollen germination. If pollen germination is not happening, it's helping us to remove that with the help of in vitro fertilization of ovary culture on stigma or the slow and insufficient growth of pollen tube as well as abscission of flowers. So all these things are possible with the help of your ovary culture. So in the right hand side, it's showing you excised flower, culture of excised ovary, and small fruit produced on the culture. So these three different functions showing you of the ovary culture that you could do with the culture of Lycopersian esculentum. So that was something about a little bit about ovary culture. Now comes the anther culture. An anther culture, it's a technique we develop anthers at a precise and critical stages that access aseptically from unopened flower bud and a culture on the nutrient medium where the microscopes within the anther develop into the callus tissues. So they help in the production of the haploid plants. Their principle behind is that without disturbing the natural habitat environment of the enclosed anther, pollen can be grown by culturing the anther. So in culture conditions, the tissues of anther will remain living and will encourage the development of pollen by nursing and providing nutrients. And furthermore, induction of haploids can be enhanced by keeping the anther of flower bud at low temperature. So this is a diagrammatic view of this is quite blurred. Let me just wait a second. So yeah, bit better. So from your flower bud, you can go into to this is your like heterogeneous diploid plant. From this, you took the flower bud, and then from the flower bud, one you could go for the pollen culture, or other side you could go to anther culture. So you gave a cold treatment to the anther culture. Then you isolated the your pollen from that. Then it goes into the pollen culture. Then multi-nutrient pollens will be there, multi-nucleate pollen, sorry. Then your prime embryo, then pollen embryo. Then further pollen embryo will give rise to your heart torpedo. Heart torpedo will give you a haploid plant. Sorry, the image is quite blurred, but if you have them in your computer, they will be fine. Then in the anther culture, you have again anther culture. You take the anther. Then further from them, you can make haploid embryo formation is happening, or the sorry, I can't read that. The further is then you have a haploid plant further from this, or otherwise you have haploid callus, then differentiating callus. So mainly the callus formation is happening on the side, and then haploid plant. So in this way, either pollen culture or anther culture, they are able to produce your complete homozygous diploid plant out of that. So importance is there is efficient way for production of haploid plants from the microspores within the anther. Production of haploid plants, production of homozygous diploid lines through chromosome doubling. Chromosome doubling is also possible, thus reducing the required to produce inbred lines, and for uncovering the mutations or recessive phenotypes. So further, let's go to the next part of callus culture. In the callus culture, callus is unorganized mass of cells, as we know, and produced from the isolated plant cells, tissues, or organs when grown aseptically and artificial nutrient medium under controlled conditions. So, plant parts are first washed with a liquid detergent, then surface sterilized by most of the chemicals such as mercury chloride, sodium hypochlorite for limited time. Then surface sterilized plant material is cut into small segments like we had discussed on the tissue culture video. If you have remember, we clean everything with the ethanol in that case, and then we cut our sterile parts into small pieces, like in that case we cut it with the cauliflower. Then explants are transferred on the suitable nutrient medium solidified with the agar. Once the growth of the callus tissue is well established, portion of the callus tissue can be removed and transferred onto the fresh nutrient medium to continue growth. Then after once your callus is grown very positively, then you can remove that part and continue to grow into the for further growth. So here you can see that in the case of carrot, you washed it, you took the phloem part, you wash them in the water, then you wash them in the sodium chloride solution, then double distill water, then you put them these phloems into the petri dish with the agar medium. And then you can see some growth started to happen, and then you take mainly the explant out of it. So this is inside is the xylem, outside is the cortex, here is the phloem, and inside is the cambium. So you just take some explant of it, including your xylem and phloem together. So it has both part of it. Then you culture into your medium, into your culture medium. Then you culture for 25 degrees Celsius for 6 to 8 weeks. Then afterwards your callus is formed. That's we have discussed before. This is how your callus culture from carrot root is done. From the carrot root, this is how you produce your callus culture. What is important? It's important for secondary metabolites. Medium moving liquid medium can be initiated from callus culture. Callus tissue is a good source of genetic variability, so it is possible to regenerate a plant from genetically variable cells of callus tissue. Furthermore, whole plant can be regenerated in large number from callus tissue. Manipulation of the nutrient and hormonal constituent in the culture medium. So these are the various importance of it. We are done with 7 cultures. Now comes the 8th culture, that is cell suspension culture. So mainly in this culture also there is a culture of a single cells or small aggregated of cells multiplied while suspending in the liquid medium. So it is also referred to as cell culture or suspension culture, we call it. The cultivation of free cells as well as the small cells aggregate in a chemically defined medium as suspension was initiated to study the morphological and biochemical changes during their growth and developmental phases. To achieve an ideal suspension culture, most commonly friable callus is transferred to the liquid medium where it's break up and readily disperse. So this is how the in suspension culture you have your lag phase first of all, then you have a logarithmic phase, then you have a linear phase for some time, then you have a stationary phase. This is the growth curve that is happening in your cell suspension culture. Remember that. And this is the flow diagram of cell suspension culture. So first, as I explained, that you need some callus tissues. Yeah, from the callus tissues you put them into liquid medium. Then further you have a shaker for that, the same. The shaker goes into cell suspension culture. Then from cell suspension you filtrate it. Then at the end you have to centrifuge and resuspend the fresh medium. You keep centrifuging it and resuspending the fresh medium into it. You re you make a pallet, then add new medium. You make a pallet, you add new medium. So by the end, then you check the cell number via hemocytometer, where you calculate in the one box how many cells are, then you multiply by 4 to understand how many cells are per ml of your medium. Then they are grown into the cell again on the shaker for some time. Then plating of cells on the cell suspension culture. Then further single cells or cell aggregates are taken out of it. Then they form embryos, and from embryos you can regenerate your whole plants. So this is how your cell suspension culture or regeneration of plant through embryogenesis is happening. It also has cell suspension, it has embryogenesis also. Centrifugation is also happening, resuspension is also happening, filtration is happening, counting of cell is happening, shaking of the cells is happening. So all these steps together make your cell suspension culture. Mainly try to remember these figures. These figures are quite important, because if you remember these figures you can explain the culture quite easily in your own words then later. So try to remember diagrams. Diagrams are important. So what are the importance of cell suspension culture? It's capable of contributing many significant informations about cell physiology, um, biochemistry, metabolic events at the level of individual cells and small cells aggregates. It's also important to understand the organ formation or embryonic formation starting from the single cell. Furthermore, mutagenesis studies may be facilitated by the use of cell suspension culture to produce mutant cells. Cell clones from the mutant plants can be raised. So, for the mutants, it's also nice to do so. So 8 cultures done. So this is, I think, the last culture, the protoplast culture, which is your cell which is without cell wall but having a plasma membrane, and fusion of protoplast is done to see what is happening, which are not able to germinate. Two different types of species. You can do it with the help of protoplast culture. So in the protoplast culture is a part of plant which lies within the cell wall, which can be isolated by removing the cell wall by mechanically or enzymatically procedure, and culture of this protoplast is known as protoplast culture. So basic culture is the aseptic isolation of large number of intact living protoplast. You remove the protoplast from your cell with aseptic conditions and culture them on suitable medium for growth and development. Then this isolated protoplast could be cultured on medium. Cell from the protoplast enter cell division and go for further cell division, which is followed by the formation of callus again. And this callus consists of property of morphogenesis and plant regeneration. So here showing you isolation, culture and fusion of fully fledged nice diagram about protoplast, leaf cell protoplast. So first, in case in A you have your potted plants, right? Then in the B we sterilize the leaf. Then epidermal peeling, we remove the epidermal layer of the leaf. Then leaf plates are coated on the enzyme solution plus osmotic stabilizer. We have to maintain the osmotic balance of the culture, so they are placed with enzyme solution plus osmotic stabilizer. Then further peel leaf segments. We peel the leaf segments further off of it. Then what we do is protoplast is sink to the bottom of petri dish. The protoplast is now what is happening after doing so much of enzyme and osmotic treatment. After peeling the leaf segments, also the protoplast just come down to the petri dish. Then plasmolized cells in enzyme mixture. when you have a plasmazide cells, then you put them in enzyme mixture. Then partial wall digestion, you remove the wall of it, cell wall of it, it's being digested. Then removal of the enzyme solution, so from the medium you remove the enzyme. Further, protoplast, we put them into the CPW13M of protoplast after centrifugation. So you do the centrifugation of these pallets, protoplast of these pallets. Then you have your pallet cell debris. On top you will have protoplast, and the bottom will be cell debris, right? Then protoplast is resuspended in the culture medium. Then you count the cells. Then further it go for yeah, whole stages of protoplast, this in to give the replating of density. So further it could go for further these reactions, so forming uh, isolated protoplast first with the P1 stage, P2 stage, P3 stage. Then wall regeneration, first they are two, then they come together, then two fused protoplast, then the wall regeneration. Then the first division is happening, then the cell clumps are happening, the clonal formation is happening, callus. Then after these so much big steps, then callus tissues started to make. Then from the callus, differentiation is starting to happen. Then the regenerated plantlet. Then at the end you have a regenerated plantlet. So this is quite lengthy, quite complicated, but at the end you have things that you were looking for from the. Then this plant could be regenerated into again whole plant. So starting from here to there, to the leaf, to the protoplast is being uh, getting down from the cell culture. Then you remove the cell wall, then you remove the enzyme, then you do the centrifugation. Then in centrifugation you have protoplast is an upper layer, then you resuspend into the medium, you check count your cells. Then you make a suspension culture. Then further within the suspension culture you put them plating of the protoplast on the plate. In the plate, further division and fusion of these protoplasts is happening, leading to your wall regeneration. Then divisions started to happen, clump of cells to happen, clonal formation is happening, then a callus tissue is forming. Then from callus differentiation you have a whole regenerated plants. So remember these figures for the pollen culture. So this technique is quite important because far-reaching applications are possible because of protoplast culture and applied in the research in the experimental biology, somatic hybridization, genetic engineering, and somatic cell genetics. So we have done quite seven number of techniques today. So these are the various applications of the same. So thank you so much. This lecture of our first lecture is finished here. So any questions? Yeah, micro. Next lecture is micropropagations. Don't worry. Don't worry. So your recording is also going fine. Students sending review articles. Sorry. Are you awake or slept already? Dun, 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 dun. So we are done with the first part. Culture media composition. Yes, yes. I want to fix one day for your practical part of view, in which I will go in details with the media's composition, which media composition is required for which one. So don't worry, in practical you will have one protocol for that, and we will go in quite detailed manner about the media. So I'm just trying to fix which day will be best for you, because first the subjective knowledge is quite important. You have a lot of questions now burning inside you, and that all will be satisfied with the questions with the practical part. So whatever you have studied, how you could use it in practical part, that's there. So, Bache, you can ask me about patents later in the later part. Let's focus on this. So I will fix now. Let's have a 10 minutes break, and we see us in the for the next lecture at 57 or so. Okay. See you soon then. Great, great, great. Just relax a bit. go through the. Okay, you can't go through slides now, but later. But in meantime, just take a break. Make yourself empty. Get some fresh air. See you soon. So class, I'm back. Let's continue. So it's successfully or is being recorded, so we can have it later also. Okay. So from your syllabus of plant biotechnology, we have covered most of your cultures. Only the last two are left. That is test tube fertilization and endosperm culture. So we deal with now the endosperm culture from all these different cultures, so that we are in continuity of our work. So endosperm culture, production of your triploid production, various factors that are determining its growth and its applications, that will be dealing in this topic. So what is history and definition? Endosperm culture is a double fertilization occurs in the majority of the angiosperms, which results in the two fusion products, that is zygote and triploid endosperms, where zygote is a fusion of your egg and one of the male gamete, and whereas the triploid endosperms are the fusion product of the polar nuclei and a second male gamete. In this, the polar nuclei and the second male gametes are united. In the first case is fusion product of the egg and one of the male gametes are united. So endosperm lack many organogenic potential and vascular differentiation. It's a unique tissue. Firstly, it's in function supplying nutrition to developing embryo, and then secondly in the being triploid. So attempt to grow endosperm culture in cultures began in 1930. Now mature and immature endosperm of various types of angiosperms have been growing. So Lampe and Mills in 1933, they were the first one to grow proliferation of immature endosperms tissue on a medium containing extract of potato. Then in 75, Nakano et al. Successfully cultured the endosperm of rice and achieved organogenesis. Furthermore, Rangaswamy and Rao in 63, they successfully grown the endosperm of Santalum album, Chandan, up to the callus. They grow up to the callus of endosperm. And endosperm culture, it is an in vitro development of isolated mature, immature endosperm. I will repeat, it is the in vitro development of isolated mature or immature endosperm from seed at a proper stage on a suitable culture medium to obtain a triploid plantlet. To get a triploid, you take the isolated mature plant from a seed, and at proper stage you grow them into the culture medium, and you have a proper triploid plant. That is your endosperm culture is, and this is the structure of an endosperm where it is present. uh, so here you can see there is a pericarp on the outside, there is a germ cells inside there, and then you have a tip cap at the bottom, endosperm lying up there somewhere, like here. So there are many different types of endosperm culture. First is the mature endosperm culture. It is a in vitro development of isolated mature endosperm, ripen endosperm of seed on suitable culture medium to obtain a triploid plantlet, and is called mature endosperm culture. So immature endosperm culture, it is the in vitro development of isolated immature endosperm, isolated precise stage from the immature seeds cultured on a suitable culture medium to obtain a triploid plantlet is called immature endosperm culture. So steps in the endosperm culture is mainly consist of three steps. First is the immature or mature seeds are dissected under aseptic conditions. Endosperms along with the embryos are excised. So from the mature seeds, you get your embryos. They are cutted out. Then these excised endosperms are cultured on the suitable media, and embryos are removed after the initial stage. Then callus formation is followed by the embryogenesis or shoot bud differentiation. It's again the same process started then in endosperm culture. Then you have a complete plant formation at the end. So here you can see initially you have some buds, then callus formation, then slowly shoot, then how it's growing the callus formation into the embryos shape structure, so into the heart and torpedo shape, so growing into the full plantlet afterwards. So further, to do your endosperm culture, you need your explant source. So in most cases of the cereals, mature endosperm does not respond to cultural conditions. Therefore, it is excised at the period of development. So when it is a developing stage, only then we excise it. Normally the endosperm of the cereal undergo certain changes 12 days after pollination, making this able to respond. Proper stage for maize is 8 to 11 days after pollination and 4 to 7 days after pollination for rice and 8 days after pollination for wheat. Then inoculation is happening. First was the explant source, which part you have to choose for. Then comes the inoculation of explant, from where you have to inoculate your explant. So for this, in vitro culture of mature endosperm seeds having a massive endosperms are decoated, sterilized with a suitable disinfectant, and after washing off 3 to 2 times, sterilized distilled water planted on the nutrient medium. Then for further, it could go into kernel surface sterilized stage. Endosperm culture is excised under aseptic conditions. And the basal white and MS media is generally used into induce callus formation in the immature endosperm. So here you can see very nicely showing you chalzal endosperm here, then chalzal seed coat. These are your suspensor. Then comes your micro pilar endosperm on this side. This is your embryo proper outside. And then this one is the peripheral endosperm. This is the general seed coat. So that's how the physiology, the general anatomy of your an embryo looks like. So the basal media that we use is suspended with the tomato juice, yeast extract, grape juice. Sucrose of 2 to 4% is used source for the carbohydrates. In some cases, we are adding 2,4-D or Indian Indo adolastic acid also cytokinin if it is necessary. So all these nutrients are also added up into your medium for the endosperm culture. Then incubation and maintenance of culture. Then you the induced callus, the endosperm cultures to induce the callus, the endosperm cultures are maintained in dark without light, and differentiation takes place of your calli at transfer to the bright light and of around 2000 to 4000 lux units and at 25 degrees Celsius. So you need this much light and then you need 25 degrees Celsius temperature. Then afterwards is the process of shoot bud differentiation is happening, embryogenesis, plantlet formation from the endosperm callus maturation, follow organogenic or the embryogenic mode of development. Embryo of differentiation occurs when the proliferated tissue is transferred from the callousing to the basal media or with or without gibberellins. Then last step, plantlet develops are hardened in greenhouse and transformed to the vermiculture media and maintain proper humidity. So what are the factors which are affecting the endosperm culture? These are explant stage, nutrient medium, how much medium you have at which stage is your plant is, physical factors like pH, and how much growth is there with the different temperature also, and for how long you're giving the photo period time for your endosperm culture. Embryo factor is also important in that case. And these are the various further the applications of endosperm culture are as techniques of endosperm culture has enabled the production of triploid plants. Triploid plants are self-sterile and usually seedless, so their trait is quite edibility of fruits and desirable in plants such as apple, banana, watermelon. So in this summer, we had most of the watermelons without seeds, and this technique, because of endosperm culture, was possible. Don't think that what we are eating seedless. Even in apple nowadays, or even guava, we see very few seeds, very little seeds at the middle of the fruit. So this is what the new plant biotechnology is happening at the local field. Further, in the timber and fuel yielding plants, triploids show better performance over the relative diploids or tetraploids. So there is no problem of seed sterility, as they can be multiplied by vegetative means. So that was something little bit about endosperm culture, not so much in details, but still I felt that we should touch this part also. So um, let's continue with the micropropagation now. This is done. So students, you will get the recorded version on this link. So maybe I should edit this. Today is day 3, right? This is series 2. So this is the link, bachcho, for further if you want to know. To save the lectures. If you want to go through the lectures, you can go through this link. So this is again we will deal with the various in this topic of micropropagation is how we done it, what are the various applications, what are the advantages, and how, various types of micropropagation also. So that will be done in this topic also. Touch with that topic. So micropropagation. By this we will be done with the unit 1 today, and then from tomorrow maybe we start with the unit 2 with the all others techniques. So in this part of unit micropropagation, I will deal with the touch shoot with the introduction, various methods of micropropagation, various stages of micropropagation, what are the advantages and commercial uses of micropropagation. Um, so it says that multiplication of genetically identical copies of cultivar by asexual production is called clonal propagation, but it's quite difficult and has been quite expensive and even unsuccessful. So tissue culture methods offer an alternative way of clonal propagation, which is properly known as micropropagation. So in this case, you can grow your miniatures of vegetative shoots within short and small period of time. Here you can see that with the plantlet, either you can have a callus or with a single node or with a leaf plant. So you take any different explant of your plant, And then from callus, either you go for shoot differentiation or embryogenesis. Both things could possible, as we have discussed quite number of times before. So then in vitro rooting will happen, and then hence you will have your transplanted to your soil system. Or with a single node, first is enhanced axillary branching, your upper part will be enhanced, then multiple axillary branching, then multiply it will enhance, then rooting media is there, then again go for transplantation. Where from the leaf explant you will have adventitious buds. From the adventitious buds you will go for the transplantation then further. So this is the basic principle of your micropropagation. So you take small part of it, any explant, and then either could go to the callus formation or single node or any leaf explant, and you have further full plant out of it. This technique has been propagated, initiated by G. Morrell, who found this very commercially viable for the orchid propagation. So orchid, the fancy plants, they are quite highly used for the same. Okay. So methods of micropropagation. Uh, it could be multiplication by meristematic tissue of axillary and apical shoots, multiplication by adventitious shoots, or multiplication by adventitious embryo formation, or multiplication through the callus culture. So all these things are possible with the help of micropropagation. So first is by axillary and apical shoots. So in this axillary and apical shoots contain most of the quiescent or active meristems depending upon the physiological state of the plant. When these shoot tubes are cultured on a basal medium containing no growth regulators, these typically develop into single seedlings called shoots with apical dominance. So here you can see there are three schemes showing you micropropagation, in vitro micropropagation by axillary bud method. Yeah, by auxiliary bud methods, upper row is applied to the rosette plant. Here is a rosette plant, and the bottom is shown to the plants which got elongated. So in the rosette plant, you have your meristem shoot tap isolated. Then shoot development is there. Then you add cytokinin to have axillary branching further. Then you have a rooting of your axillary form shoots. Then on the other side, at the bottom row, you have a meristem or the shoot tip isolated in this case, or node is isolated. Then other further shoot is developed. Then after the shoot development, we add cytokinin for axillary branching. Then after the axillary branching, you are rooting of your axillary form shoots. Then you can transfer to the complete plantlet. So this is how you do it with the axillary bud method. So this is shown you in vitro micropropagation of asparagus. It's used as a very expensive vegetable and eaten as a boiled or a saute form. So this is how the asparagus is being grown like this, showing you in vitro. On the contrary, when the shoots of the some explants materials are grown on the culture medium containing cytokinin or axillary shoots develop precautiously, which proliferate to cluster of secondary and tertiary shoots also. Then comes with the adventitious shoots, multiplication by adventitious shoots. First was the axillary buds. So axillary buds are performed meristems present at the leaf axils, whereas the adventitious buds may arise from the any plant structure, and this regeneration is also dependent on the presence of the organized plant tissue. So explant maybe in this case would be stem, internodes, leaf blades, cotyledons, root elongation zones, bulb, corms, tuber, rhizomes, or etc. So high number of adventitious shoots may arise only from a single epidermal cells. And continuous propagation of adventitious shoot proliferations from bulbs and corms can be achieved by cultivating two vertically spread pieces of shoot bases. Now, after clearing with the axillary bud and adventitious shoots, now comes the adventitious embryo formation, the third method of micropropagation. So, in this case, it is another useful approach followed for many important plant species. An adventitious embryo can arise directly from the group of cells or within the original explants or from the primary embryos. So there are many plant species which develop embryos in vivo from diverse kind of explants. That is, orchid leaf tips produce large number of embryos, whereas citrus and mangifera have shown to develop polyembryos from the nucellar tissue. So these adventitious embryos are deployed in nature and can be used in the clonal micropropagation. Fourth one, the last one, through the callus culture. So in this case, direct plantlet formation from the explant in the culture is from the desirable in the case of a micropropagation or clonal propagation. But shoot formation may occur through the organogenesis or embryogenesis from the callus produced from the explant. So in this case, from the callus, you can do the organogenesis or embryogenesis to do your micropropagation. So this is also one way to go for. But one disadvantage is that the callus cells are not genetically stable, so it cannot be single clone, and this process is more time consuming. Also, plant regeneration capacity may decline due to the genetic unstable condition. So, so in some cases, plant species has been genetically stable calli have been produced. Calli is a plural form of callus. So, in these type of calli, shows meristematic zones are formed from the peripheral layer. So, the meristematic layer invariably comprise of diploid cells expressing totipotency, and these type of calli can be subdivided into smaller parts and which may produce multiple shoots from each other. So we are done with the various methods. That is, first was with the axillary bud, then the adventitious shoots, then the adventitious embryo formation, then the fourth was the callus culture. Now comes the five stages of micropropagations. What are the five stages? So initially, first in the first slide, we saw the just summary of it, the micropropagation, how it is done in a very easy manner by using different explant part of your plants. Now we will see how it is done practically with each stages. So with the stage 0, you have a preparative stage. So this stage involves the preparation of mother plant. Your mother plant is prepared before you go to the aseptic culture of stage 1. And also to reduce the contamination problem in the subsequent stages, mother plant should be grown in the glass house and watered so to avoid overhead irrigation. This will also reduce the need of harsh sterilization treatment. And in the stage 0 also include exposing the stock plants to the suitable light, temperature and growth regulator treatments to improve the quality of explant. Or in case of photosensitive plants, it may be possible to obtain a suitable explant throughout the year by controlling the photo period in the glass house. So you can maintain the photo period time, you have to maintain light, temperature and growth regulators, and you have to maintain the glass house and the water treatment so that the irrigation could be maintained and proper light could become to your plants. So please maintain all these features before starting your micro propagation. So So to initiate your culture, first stage one is the your explant, taking your explant, which is a virus-free plant without any infection. It should have to start it. It should have some millimeter of shoot tips. If the stock is virus tested or virus eradication is not necessary, then most suitable explant is nodal cutting. You just do the nodal cutting to do so. Small shoot tips explants have low survival rate and show initial growth. So furthermore, sterilization has to be done of your parts of the cuttings to in a greenhouse. Discharging the surface tissue from the plants while preparing the explant also minimize the loss of culture due to the microbial contamination. Then the third part in the stage 1 of your sterilization, after the sterilization is browning of the medium. A serious problem with the culture of some plant species is the oxidation of phenolic compounds. Your phenolic compounds. could be oxidized within the cell surface. It could be leached at the surface of X plant. So it turns the medium dark brown and is often toxic to the tissues. So this problem is common from the adult tissues from the woody species. From woody species, that's quite common in that case. Now, after having the stage one of initiation of the culture, having selected the plant with virus-free, sterilized, and also check the browning of the medium is happening or not, then you go for the stage two with multiplication. And this is the most crucial step, which most of the failures are happening. Basically, we take three broad approaches to follow to achieve in vitro multiplication through callousing. First is through callousing. The potentiality of plant cells to multiply indefinitely in cultures and their totidependent nature permits a very rapid multiplication of several plant types. Differentiation of plant from culture cells may occur via shoot root formation or somatic embryogenesis. So differentiation of plant could be done by organogenesis or by the somatic embryogenesis. Whereas the somatic embryogenesis is the most appealing from the commercial point of view. As once established, it will lead to the better control than the organogenesis. Since somatic embryos are bipolar structures with defined root, shoot meristems rooting stages are required for the microshoots got eliminated. Then, further, somatic embryos being very small and uniform bipolar in nature, they could be they could be stored in the cold storage or cryopreservation, or remove the water from them by doing a desiccation for prolonged periods. So you can keep them for the long period. These by callousing. So this is shown you the whole stages in a very nice diagram, everything what we are studying so far. In the first stage is the establishment of your X plant in the culture. So you have your X plant source, then you maintain the medium with the organic elements, organic components, carbon source, growth regulator support. So with the all four main ingredients in it, you have a medium prepared, then you maintain the environment with the light and photo period and the temperature. So these things are maintained for your explant. Then in the second stage, multiplication of your propagules. So whatever the plants that you have selected, then they will multiply within this with the help of if your auxin is to cytokinin ratio is less than 1, then you will have shoots or callus formation will happen. So like this, you change the hormones concentration and grow as it is. Then comes the stage three, that is pre-treatment for transfer to soil. So your plants are pre-treated for transfer to soil with that, and then root formation in vitro. Then you add auxin to cytokinin ratio to be more than 1, so that the root formation starts in vitro. Then furthermore, axillary shoot formation, adventitious bud formation, somatic embryogenesis. So by this method further you grow your whole plant later on. So this is shown you the how micropropagation is done in detailed manner now. So I hope this statement, this the definition and its steps, various ways of doing it and how it is done might be cleared with you. So yeah, so we were doing with the multiplication, we are done with the callousing. Now comes with the adventitious. You can do it with the adventitious, but also micropropagation. So in this, you used the apex part, shoot apex part or leaf axil part of a plant which is known as adventitious buds for growing it, and the shoot differentiated from calli should be treated as adventitious buds in this case. So it has been done in the case of many crops, from the roots of blackberry, raspberry, leaf of crassicula cuttings is standard horticulture practice. From the bulbous plant, that is lily, adventitious bud formation is the most important for mode of multiplication. So third is you can also do it with the help of enhanced axillary branching, where you have to take care of the suitable cytokinin concentration with or without auxin, and due to the continuous availability of cytokinin, the shoot formed by the bud present on the explant and develop axillary buds, and hence you have mass of branches afterwards. Now, as we discussed already, two stages multiplications. Now comes the transfer to the soil, that is rooting of your shoots. Then this is done in somatic embryos carry a preformed radical and may develop directly into the plantlet. However, these embryos are often very poor in conversions into plantlets. So what we do, we have an additional step of maturations to acquire the capability for normal germination. So adventitious and axillary shoots develop in these cultures in the presence of cytokinin generally lack roots. To obtain full plants, the shoot must be transferred to the rooting media, which is different from the shooting multiplication medium, and particularly in its hormone and salt composition. Then comes the last stage, that is transplantation of your propagated, and it's depend upon the different plants that you have to transfer. So two main deficiencies in vitro grown plants are the poor control of water loss and heterophytic mode of nutrition. Therefore, a gradual acclimatization is necessary. So they have to acclimatize to the new atmosphere when you are adding, so that they do not get destroyed after done with so much of experiments with them. So proper greenhouse field has to be taken care, and during acclimatization, in vitro form leaves do not recover, but plants develop a normal leaves and functional roots in that case. So after the transplantation, this is shown you of V. retici, one plant, how the micropropagation is done. You taken the donor plant, seen details here. And then this is the yellow cluster of the induced MS medium free of PGR. This is in the yellow modular cluster of culture induced with the PGR. Then these are the nodules cluster subculture in the MS culture medium suspended with the GA2 isolated in the high proliferation rate and subsequent development of the adventitious microgrowth. Then further in the D we can see the elongation and the growth of the shoot in the MS culture media free of PGR. So they are growing in the MS culture media which is free of PGR medium. Then acclimatization of the plant, that when they are going to the new field they got acclimatized nicely. Then growth in the greenhouse, then the growth is happening in the greenhouse further. So this is how, this is quite complicated all in all this technique, but it's being highly used in various orchard plants and also in vegetables and in other fields also. So this is shown you one other examples how potato propagation and barley propagation is done. So here you can see in the bottom uh, that first you take a potato sprout from you have a potato plantlet out of it. Then you do the internoding sections. Either you take the leaf disc or you take the petiole section or the root section. You can take any of these sections and then go for the culture medium. Then from them you have shoots from the internodes. So shoots are growing from the internodes, and then potato plantlet is coming out of it. Then the mature potato plant is growing. Or potato tuber disc, from the tuber plant you have tuber disc coming out of it. Then shoot generation is happening. Then potato plantlet is coming. Then you have a mature potato plant afterwards. And this could go further into back thing. So this is how you propagate your potato. So this is micropropagation of potato showing to you. On the other side, on the top one is the barley propagation, where you have a barley seed heads. You take embryo or embryo without access, then you put them into the culture medium. So in the one with the embryo, you have the callus from the embryo, then shoot regeneration and root planting. Then it could go to the mature barley then further. And this is the barley micropropagation of barley shown to you in summary how it is done. Okay. So what are the advantages of micropropagation? Uh, it's a technique which has helps to enhance the rate of multiplication. It has helps to give us million of shoot tips can be obtained from this technique within a short period of time. This technique has helped us usually a short cycle also, usually 2 to 6 week, with which we can result in the logarithmic increase in the number of shoots. Bulb or comb producing plantlets are possible to grow with this technique. Storing and transporting takes lesser space. Soil-free environment is possible. Stocks of germplasm can be maintained for many years by these methods of propagation. You have disease-free propagules are also possible. Pathogen-free plants are produced in this case. Further, minimum growing space is required as we discussed already. So culture in the culture vials within the lab, you can grow all those so many plants within the small period of time with the help of this technique. And furthermore, new tissue culture are obtained through the DNA technology or haploid culture hybridization source of tissue material. So what are the commercial uses? They are mainly micropropagation of orchids are quite famous. They have a lot of profit in the world, commercially exploited around with the chrysanthemum, carnation, gerbera, anthurium. So all these have been growing because of this. A large number of forest trees, oil producing plants, vegetables, many edible crops are grown with the help of this technique. So this is shown you how micropropagation done in the different plants, the bottom one in this case. Further, what are the commercial uses of micropropagations? 100 woody species are been into this part of it. It is large scale, cost effective, nursery friendly technique it is. It's been published, methods have been published quite number of in general so that everyone can use it worldwidely, so you don't have to follow your own methods. Bioreactors are being used for to multiply osmotic embryos, shoots, and bulbs into the higher amount. So automatization is also being done in the subculture of the stage 3 and 4. So they are done automatically, done by the machines. And so this reduces the cost of labor component in micropropagation. So there are huge number of advantages as we can see. So thank you very much. That is for today. And last, before we leave, let's what we have studied. It was quite number of things we have studied suddenly. So for you, it will be hard to absorb at once. To make it more clear, let's have one video. So a scientist is showing us how it is done. So I will not speak when video is going. So be concentrated and go and keep your ears open then. Yeah. So I will start.
Controversy lately about human cloning and related issues like test tube babies. But have you ever heard about plants in test tubes? Welcome to the world of plant micropropagation. This is the last of the asexual propagation methods or approaches that I'd like to talk about in this cyber tower room. Micropropagation is a rather general term that refers to any of a number of different approaches to plant tissue culture where the objective is to multiply plants, to bring about propagation. All these different methods have in common three features that are characteristic of most micropropagation systems. First of all, you start with a small explant. The image on the right of the screen shows a very small apical meristem. It's only about 1/10 of a millimeter in diameter. That's the kind of explant that can be used to start a plant tissue culture. Second characteristic is aseptic culture. We work in a germ-free environment where we exclude microorganisms, and the only living part of this system is the plant itself. Because of the sugar in the nutrient media, microorganisms would overtake the entire system. And the third characteristic is heterotrophic nutrition. In other words, these plants are not dependent on photosynthesis for production of sugars, but instead we provide the sugars in this artificial gel nutrient medium along with mineral nutrients, vitamins, and plant hormones. So I'd like to show you two different micropropagation systems. One is organ culture, and the other is somatic embryogenesis. Organ culture depends on proliferating shoots in culture and then using hormones to induce adventitious root formation. So basically we're dealing with cuttings at the end of the process, but they're micro cuttings. somatic embryogenesis is a variation on the theme we considered earlier of asexual embryo formation. So let's go over to the sterile work area and take a look at it. Well, okay, in the case of organ culture, or shoot organ culture in this case, the strategy is to take a small explant, either a actively growing shoot or even a dormant bud, surface sterilize it so the culture remains aseptic, get rid of the bacteria and fungi that are on the explant, and then place that bud into a culture medium that contains a hormone called cytokinin. That cytokinin stimulates branching of that shoot, just as if you had pinched it back to induce lateral branches to form. So basically it turns into a very small bush or shrub-like structure, which we subdivide, place on fresh medium, repeat the process, and increase as much as we need to. You can see here that if we have a 5-fold multiplication rate every 6 weeks, that will go through 9 generations, about a year's time, and come up with about 1.9 million plants. There is no other asexual propagation technique that has the potential for that extremely high rate of increase. Now, let's take a look at how it's actually done. I need to grab a petri dish up here. This has been pre-sterilized. And this is a culture box that has been initiated already. And what we're doing is going through one of those rounds of multiplication. Use the forceps here. I have to surface sterilize them in this red hot back incinerator for about 10 seconds. Allow them to cool for just a moment. And then reach into the culture vessel. Use them to pick out one of those clumps very gently. Can't touch it with our fingers to avoid contamination. Lay it on the surface of a sterile petri dish. Sterilize the scalpel. And use that to cut away the callus. And basically divide that clump into a number of individual shoots. There are 5, so that's a 5 or 6-fold multiplication rate. Then we can take the individual shoots and transfer them to a fresh nutrient media, once again containing the sugars, mineral nutrients, and the hormone cytokinin. Take any one of the shoots and place it into the culture vessel. We can put about 9 shoots into a vessel like this. And we get the requisite number of shoots in there. Close it up. Seal it off so it maintains aseptic conditions, and place it on a lighted growth shelf to grow on for another 4 to 6 weeks before we subculture it again. And so, once these microshoots have multiplied through several successive generations of subcultures, we have many, many shoots, as many as we need, perhaps 1.9 million if we need that many. But these are shoots without roots. And so what we have to do is induce adventitious root formation on those shoots. We do that by either placing them on a fresh nutrient media that contains the rooting hormone, auxin, or we can take these microshoots out of culture and place them up in the greenhouse under a special fog tent. And they will basically root just like a cutting that we discussed earlier, except on a very, very small scale. And we'll wind up with rooted microshoots that can be grown on into full-size plants. The next micropropagation technique I'd like to talk about is somatic embryogenesis. This is regeneration through induction of asexual embryos. We talked about how it occurs naturally on the margins of leaves and in the case of clonal seeds called apomixis. In this case, we're using a sterile micropropagation system, and this happens to be a cluster of callus and somatic embryos of American ginseng, Panax quinquefolium. We're very much interested in this plant in our research program. As it turns out, American ginseng is normally propagated from seed, but it is extremely difficult to propagate clonally. It has a single shoot that comes up from the ground, only one determinate shoot with 3 or 4 leaves on it and a small flower cluster. It will not root from cuttings. You can't propagate it effectively by division. Grafting is not an option. And so the only approach that has been even marginally successful is micropropagation. What we do is we take a ginseng root. You can see in the photograph here, it's a thick, fleshy root, highly valued as a medicinal herb, as a matter of fact. We take that root and surface sterilize it, cut it apart, and take a small chunk of tissue out of the inner portion of that root and place it on the appropriate nutrient media containing a combination of plant hormones, auxins, and cytokinins. And these induce the cells from the root to begin to divide in an unorganized fashion and give rise to a callus, similar to the callus that we talked about in the case of graft union formation. And then by changing the nutrient media to a different combination of hormones, that callus can be induced to form somatic embryos. And these are basically very similar in structure to normal sexual embryos, but they are occurring under these plant tissue culture micropropagation conditions. The thing is, they do not have a regular seed coat like a typical sexual seed does, so they have to be handled very, very gingerly. There's a procedure for getting them to germinate in culture and then finally acclimatizing them so we can get them out of culture. So let's go back to the culture room now, where we grow these plants on under artificial lights. Micropropagation, then, is an important tool of the plant propagator in modern horticulture. It hasn't displaced the other forms of propagation that we've discussed, like some folks thought it would 30 or 40 years ago, but it is a very important tool used commercially to propagate such crops as rhododendrons, mountain laurels, and many of the houseplants that you have in your home. Now let's turn to the issue of the role of micropropagation in modern biotechnology. In addition to being used as a propagation tool per se, micropropagation is an important tool for plant breeders. Those molecular plant breeders involved in genetic transformation or the production of genetically modified organisms. Now, genetic transformation of plant cells is a different issue in some sense. It has to do with transferring genes from one organism, such as a bacterium, into another organism, such as a plant. And that's not micropropagation per se. But where micropropagation comes in is that once you have transformed those cells, then they need to be regenerated into whole plants. And that's either done through somatic embryogenesis or adventitious organogenesis. Now, I'd like to just briefly talk about an issue that is confusing in some people's minds. We hear a lot of talk about human cloning and so forth, and we're also concerned about issues related to genetically modified plants or genetic engineering. There's a tendency for some people to confuse the two. In fact, genetic engineering utilizes micropropagation, it utilizes cloning, but the two aren't the same thing. As you can see from this article in the New York Times, there is a tendency for even reporters at the New York Times to confuse the two. This is really an article about genetically modified organisms, but they talk in this particular highlighted sentence about as if cloning were the same thing. And so you see, regardless of what you might think of human cloning or cloning of animals, the cloning of plants is a very important part of modern agriculture. In fact, a part of agriculture ever since the invention of agriculture 12,000 years ago. Well, I hope I've piqued your interest in plant cloning. I'm sure you have questions, and if you do, I welcome you to ask them in the discussion board I'd be happy to respond to them. If you'd like more information about plant cloning, there are many books that I could recommend. And also, I teach an online course in grafting. It's called The How, When, and Why of Grafting. You can get more information at the URL that you can see here. Thank you.
So, this professor has explained micropropagation and somatic embryogenesis quite in detailed manner. So both are. It's not, as I also explained, that some people get confused. Uh, you get confused with the term of human cloning and cloning of plants. They are quite different, and it's highly being confused even by the New York reporters that might be the same. So that's why some people really don't eat GMOs, and they hate to eat to get your plants to be uh, genetically modified, and you're eating them. So so that's how it's quite varied technique that they were telling us. So um, um, yeah, so take home lesson for today is that we have studied about various tissue cultures techniques, from in vitro ones, from the callus culture, organ culture, all those suspension culture, uh, many techniques, protoplast culture, um, and then we went for to the endosperm culture also, and then we went for micropropagation and how various stages of it, various methods of doing it, and all those things. So at the end, the professor just explained us how it is done by removing the shoots, then growing them into the agar medium with the help of oxygen medium to create roots, or just directly shifting them to the greenhouse. So that was the main things that we have discussed all in all today. So you can access to the lecture of today uh, via this link. I will share it here also. Uh, so you can share it later. So now your questions. So as we discussed, they are quite different. In micropropagation, you have different stages to go for, and it's quite particular for different plants. It's not happening in all plants, whereas the tissue cultures has wide variety of different cultures you can do it, like 9 types of we've discussed. So both are quite different. So yeah, that's true. That's also true that it comes under the tissue culture, but it's a whole new chapter. It's a whole new chapter. Gaurav, you always surprise me with your questions. I really like your intuition power. You will be a great scientist, better than me, I'm very much sure. How will the specify the amount of browning preventing agents to implement the culture media with respect to its concentration? Oh, I have to go through it. I'm sorry, I don't have answer to it, but I will get back to you about this tomorrow. So I will copy your question in my notes and get back to you tomorrow. Uh, so, okay, class, that's it for today. Uh, um, so, I will see you tomorrow. Maybe we will touch the practical part of it more tomorrow. Do some uh, medical practical part to touch it from the syllabus itself, and continue with the lectures also. So, we do 2 parts tomorrow then. Yeah, in meantime, go through the, I will send you the lectures also just now. Just stay there so that I do not forget later. Plant biotechnology, day 3. So, there you go. So you will receive all the lectures of today and the video also soon into the group. Okay, then I wish you all a good night. Take care, and see you tomorrow. Same time, same place, same, uh, yeah, Google Meet. I was supposed to do it actually, but you will also have no link of the YouTube. So please subscribe to the channel also, so that whenever we are online, you can come and see it, the lectures. So I wish you all good night. Take care. Bye bye. Bye. Thank you so much. You're welcome. You're welcome. So I will leave off now. Bye then.